Hello, everyone, and welcome to the ICFJ Global Health Crisis Reporting Forum. My name is Paul Adepoju, and I'm glad to introduce our experts today. And today we are looking at an issue that is at the core of the current response strategies to the COVID-19 pandemic. And that is looking at the COVID-19 vaccine gray areas. Quick background, if you're following developments in various parts of the world, especially around last week, we heard the news of in Europe and even in several other countries where the COVID-19 vaccine rollout plans were suspended because of news surrounding one of the vaccines being associated with blood clots. And there are several, while towards the end of the week, we began to realize that the number of cases were so few to warrant shutting down entire vaccine efforts. And we've been hearing lots of information, a lot of details, and different insights about issues that are affecting and impacting COVID-19 vaccines in general. But today, we have an expert, Professor Lorenz von Seidlein, who is an associate professor at the Mahidol Oxford Tropical Medical Research at the Faculty of Tropical Medicine, Mahidol University in Thailand. And he'll be sharing a lot of information and will be enlightening our audience regarding how vaccines work, why they are important, and how COVID-19 has impacted vaccine development. He will also be talking about lessons and implications for other diseases, especially one of his research interests, which is malaria. And if you are joining us from Zoom today, please submit your questions on the Zoom chat, which you will find in the menu option below. And if you are watching from the Facebook live stream, I advise you to post your questions in the comments below. And so without wasting so much time, I'd like to introduce Professor Lorenz. Professor Lorenz, how are you doing today?
Hi, good evening. Thank you very much for having me. Good. So let's get started.
Are you ready for our journalists?
We are ready.
Yes, over to you from there.
Okay, so what I would like to talk about is COVID vaccines. And there I would like to specifically talk about vaccines made in the USA and in Europe, and vaccines made in China, India, and Russia. Then talk a little bit about the challenges for the next vaccine trials, for the next wave of vaccine when that comes along. The usual questions about coverage, herd immunity, how much coverage do we need to interrupt transmission? The COVAX facility, then the emerging variants and the spread of variants and what that means for vaccines. Touch quickly on vaccine hesitancy, and then at the end close off with the transformation following the pandemic. I was asked what are the effects of the pandemic on vaccine development as well as other diseases, like for example malaria. So start at the beginning. What is a vaccine? A vaccine resembles a disease-causing microorganism which is recognized by the immune system and then triggers an immune response, which consists out of two parts. One of them are the antibodies, neutralizing antibodies, about which we will talk later about, and the cellular immune response, about which we will not talk today. And then the next question is, once you have a vaccine, how is it evaluated? And this is critical to understand. So once a vaccine has been found potentially effective in an animal model in the laboratory, it is usually put into a phase 1 study. And a phase 1 study, a small study in healthy human beings, usually young men, maybe 10, maybe 100, somewhere in that range, to see whether this vaccine is safe, whether there are any kind of big problems. And once that is passed, then the vaccine goes into a phase 2 study, which can be maybe 100, 200, 300 volunteers participating in that. And in the phase 2 study, we try to find out whether the vaccine is safe again. And secondly, whether it's immunogenic. You need more participants to take part in the study to find out whether it really produces the antibodies that we are looking for. And when these two phase 1 and 2 have been passed, then the vaccine goes in the phase 3 study, which is usually pivotal, meaning that if the vaccine passes this stage, it can become authorized. So for the COVID studies, there's an agreement with the regulatory agencies that the vaccine should, that in the phase 3 study, there should be about at least 30,000 you need at least 30,000 participants for a phase 3 study. And if that's successfully passed, then the vaccine gets, in a pandemic like now, an emergency authorization. And then from there on, it gets a full license and approval. During the emergency authorization, the vaccine producer, the vaccine company, has to report on every type of adverse event that comes along. So there is a very strict network of pharmacovigilance which detects anything that happens to people who have been vaccinated, basically. That is reported, that is filtered, that is examined, and then there are the next steps happening. So the number of that you see here, the numbers this here, there's a thing. Let me just show here. Here, these numbers are quite astonishing. So there are more than 100 vaccines, or nearly 100 vaccines, reaching now phase 3, and there are already another 13 which are out there, approved and available vaccines. And that all happened within 1 year, so it's very, very quickly. And another thing that I find extremely unusual about this is that only 4 vaccines during this process have been abandoned. Usually the ratio is the other way around, that there are much more abandoned vaccines than vaccines that are progressing. With COVID vaccines, it's very unusual, very fast, and a lot of successes. Oops. All right. So let me quickly talk about the different types of vaccines that we are dealing here with, because I think that's also very important to understand what are the vaccine platforms or vaccine classes that we are dealing with here. So the first one is the mRNA vaccine. This is messenger RNA. And here this messenger RNA molecule in the lipid coat is injected into the vaccine. And it is imported into the cell, replicates in the cell, and then produces protein. The protein is released from the cell, and that triggers the immune response. It's a very basic process. A similar way of doing it is vector vaccine. Here it's not directly injected, the RNA, the genetic material. The genetic material is spliced into a vector, which is usually here an adenovirus, which is completely harmless to the human being. And that again is imported into the cell, replicates, produces protein. The protein is released and triggers the immune response. Here, this is the vectored approach. These two approaches have not been around in the last century. Vector vaccine may have been around maybe a little bit more than 10 years. mRNA vaccine are something completely new that has not existed before the pandemic. Nobody has really kind of had a functioning mRNA vaccine. And the surprising thing is so that it's completely new, that it was very fast, and it works, and it is safe. So for us who are people who have worked in vaccines for a long time, that sounds a little bit miraculous, because a year ago, very, very few people would have believed that mRNA vaccine can actually do this, but it is working. And there is very close, as I tried to say, very close pharmacovigilance, trying to follow people who have been vaccinated and see whether they run into problems, whether there are unusually large amounts of anaphylaxis or any other problem. And that has really not been found with the mRNA vaccines. We talk about the vector vaccines later. Another vaccine type then is the subunit vaccines. And here what you do is you just take a part of the organisms. Here, if this is meant to be this kind of schematic drawing of a virus, here is you have these spike proteins, and you just take these proteins, conjugate them to something to make them a little bit better recognizable by the immune system, and inject them. So the protein is directly injected into the human body, combined with an adjuvant to trigger a better immune response, and that causes then the immune reaction. So that's a method which is around maybe for 40 years. It's unusually safe because it doesn't have any replicating parts in it, and it does not have any live virus, or it didn't come in touch with a live virus. So that was a very popular way of making vaccines in the past, but probably that will be now surpassed with newer methodologies. And then finally, we have the oldest way of making vaccines, and that is an inactivated vaccine. You can inactivate the virus, for example, using formaldehyde or other chemicals, or also you can even heat inactivate the virus and then inject the dead or inactivated virus. It's an old method. Sounds a little bit like a crude method. The huge advantage of it is that you use the entire virus. So there are plenty of targets for the immune system. If we use a subunit vaccine or even an mRNA vaccine, we produce only a single protein. So that's the spike protein usually in COVID. And they're a certain domain, the receptor binding domain that we are particularly interested in. But if we use an inactivated vaccine, we have a lot more targets. So in theory, an inactivated vaccine could perhaps be more robust against variant viruses. So these were the platforms that I was trying to show you here. And then we have here the vaccines that are really in use today, that with the ones we were talking about, 4 vaccines plus a 5th one, which is coming out now and is very strong. These are the vaccines that are in the headlines in Europe and in the United States. So we have here the starter is again this mRNA vaccine from Pfizer, BioNTech, that provides, that was the first one out, had a protection of 95%, very strong. It is reactogenic, so when you get vaccinated, people feel sometimes a little bit under the weather for a day or two, but there are no stronger complications associated with this vaccine. A disadvantage of it is that it needs the storage as -70 degrees. They are working on it, trying to bring it, make it a little bit less strenuous. But at the moment, I think it still needs -70 degree for storage. The other one is Moderna. It's basically the same, also 95% protection, but it can be stored at -20 degrees. That's an advantage. Then we come to the AstraZeneca Oxford vaccine, and that is a little bit more complicated. I'll talk about this later, but again, has been now approved in many countries. 2 really immediate strong points of this vaccine is it's very affordable. It's cheaper than the mRNA vaccines, and it can be stored at 2 to 8 degrees in a basic fridge. Then we have the Jensen Jnj Vaccine, which has in different trials had protection between 57, 72%. Again, it can be stored at between 2 and 8 degrees. One of the strong points of this vaccine is that it needs only a single dose. The others need 2 doses. And then finally, Novavax, which is not out yet, which has not been approved yet, but we expect that it will be approved in the coming weeks. It is perhaps the strongest of this vaccine. It's a subunit vaccine, so it's very safe. It gives similar protection as the mRNA vaccines, and it can be stored in a normal fridge. So these are the vaccines which people talk about when they talk about vaccines in Europe and North America. Allow me to say a little bit more about the AstraZeneca vaccine, because it has been so much in the news. So there is a big interest in this vaccine. So this vaccine has now received regulatory approval in more than 100 countries. 20 million doses as of today have been administered in the United Kingdom, and 27 million doses have been administered in India. So as vaccine developers usually say, once you have the third 3 million doses are out, you can be relatively sure that the vaccine is safe. These vaccines here is with 47 million doses administered. You can be pretty sure that this is a quite safe vaccine. As I said, it's affordable, it's robust, and it's quite well suited for low and middle income countries, which is the focus of our talk today here. The controversies of this vaccine are starting with the first trial, which was conducted in Brazil, South Africa, and United Kingdom. The data from Brazil and United Kingdom were included in the first publication. The problem there was that they started off with giving a half dose to the first 5,000 participants, and then delayed the second dose. The result of that was that when they resulted the results, and that was only 3 months ago, I just looked it up in last December, basically they had 3 groups or 3 results they were reporting on. The first one was the people who got the standard full dose, and then a second full dose, and then were evaluated. And in these people, the protection was 62%. And then they went back and analyzed only the population, the people who had received the half dose and then with a delayed second dose. And they found that in this population, the protection was 90%. This was, however, a very unusual subgroup in that the participants were under 55 years of age. So they are not generalizable. They are not, maybe have a different immune response from older people. And then they did a third analysis where they combined the two groups, the one who received the half dose and the ones who received the full dose. And so they had basically here 3 assessments for the same vaccine, one with 62%, one with 90%, and one with 70%. And that was confusing. Even for people who have spent a lot of time thinking and working on vaccines, they were confused by that. Then they had the redemption trial, so to speak, which has been only released earlier this week. That was a trial conducted in the United States, Peru, and Chile. And they had, the first press release that came out last weekend was here this 79% vaccine efficacy. And within 24 hours, the DSMB, the Data Safety Monitoring Board, which is basically monitoring the data that are coming in and is assuring safety, said, well, we don't believe that. We think that the true result is somewhere between 79 and 74%. And as I'm speaking earlier today, Astra Seneca turned around and said that they now believe that their vaccine efficacy is 76%. Whether it is 76% or 79%, it really doesn't make a big difference. What it does is that people are, again, not totally comfortable, because I think most people would just like to hear, this is the result, that's it. That happened for the Pfizer vaccine, for the Mrna vaccine. It would be just nice to get one single result and be told what is happening. That is problematic with this vaccine. Then we have the problems with the EU, the European Union. The European has a central regulatory agency, which is called EMA, the European Medical Agency. And that EMA has approved the vaccine and has continued to approve the vaccine. In contrast to the United States, where the US FDA has not approved the vaccine yet. They are basically waiting for the data from the trial in the Americas. Then earlier in March, there were some reports about blood clots. They were first reported, I think, 3 blood clots from Norway, which were investigated because, as I was trying to explain earlier, once this vaccine received this emergency authorization, there's this very strict pharmacovigilance. So this picks up adverse events like, for example, clots combined here with thrombocytopenia. And as we are speaking now, the rate of these clots and thrombocytopenia following vaccination is about 4 per million. And the complicated thing there to understand is that there is a background rate. These clots and thrombocytopenia happen all the time. So if you vaccinate 40 million people, you can be sure that some of them are also going to get this disease. And it is the understanding today that this rate of clots and thrombocytopenia is not higher than what you would expect as a normal background rate. But nevertheless, Austria first suspended the use of the vaccine, followed by Denmark and Germany. And there's then suddenly this mechanism going on that the governments want to make sure that the population can trust them. And they say, okay, so if we are in doubt, we better suspend it and stop using it. It demonstrates that pharmacovigilance works, but it causes considerable confusion if you have a vaccine going out and then you stop it. And as far as I know today, the vaccine is still suspended in several countries in the European Union. This is a particularly unpleasant situation for people who have already received the first dose and now are unsure what happens about the second dose. But please allow me to say this is an affordable, robust vaccine which affords good protection. People who are working a lot on vaccines still believe this today. Okay, so that was the Oxford vaccine. Then we have 2 vaccines coming from India and onto the international market. And the first one of that is from the Serum Institute of India. This is exactly the same vaccine as the AstraZeneca vaccine. It's the same construct, so it has the same characteristics. The difference is that it is produced in India, and not many people know this, but the Serum Institute of India is by volume the largest vaccine producer in the world. So they have it relatively easy, or easier than others, to scale up to make hundreds of millions of doses of vaccine, and that is what they have been doing already. So they're contributing huge amounts of vaccine internationally. The other vaccine is from Bharat, again, an Indian company which makes huge volumes of vaccines. This is an inactivated vaccine, provides about 81% protection and is being rolled out at the moment. Then, of course, we have the Gamaleya, the Russian vaccine, which had a little bit of controversy in the beginning, but they have published a complete phase 3 study in the Lancet and showed that the vaccine produces about 92% protection. This vaccine is rolled out in also internationally. And then we're coming to the really important vaccines coming from China. For low and middle income countries, they are the major players. And there are currently 4, or if you want to count them as 5, vaccines coming from China. The first one here that I would like to mention is a Subunit Vaccine from Seidlein. This vaccine has been approved in China earlier in March and is now being rolled out. Unfortunately, we have no data from phase 3 trials. We do not know the protection of this vaccine. The internal data from the company suggests that it's a very strong vaccine, provides good protection, but it's really only rumors. The other vaccines are Inactivated Vaccine, probably the most highly distributed one, which is available throughout Asia now, is the Sinovac vaccine, which produces 57% protection, and CanSino with 65%. And finally, Sinopharma, which is probably provides close to 80% protection. These vaccines can all be stored in a refrigerator. They don't need special freezers. And let me say a little bit more about Chinese vaccines, because they are so important in Asia and other low and middle income countries. These vaccines have all been through phase 2 studies, and the phase 2 studies have been published. None of them had a phase 3 study published so far. Important background information is that China is building at the moment a vaccine production facility with a capacity of more than 600 million doses per year. If I understand this correctly, this is for Sinopharma. Another interesting fact is that China is exporting more vaccine doses than it is currently using within China. So the large majority of this vaccine is actually exported. There have been questions about WHO pre-qualification. So the WHO pre-qualification, this is a mechanism which allows the evaluation of products to be bought in the UN system. For example, UNICEF can buy only products which have this pre-qualification from the WHO. And Sinovac and Sinopharm have already applied for this and should probably get this approval in the coming weeks, because they have in the past had WHO pre-qualification, so it's not such a big step for them. There has also been criticism of China in the past that there's only minimal involvement with the COVAX facility. By now they have donated 10 million doses, or pledged to donate 10 million doses. There are several reasons why people are criticizing the Chinese vaccine producers. One of them is that these are bilateral deals, so between one country and a Chinese company. All of these aspects of vaccine production, even vaccine evaluation in China, has to be government approved. So there is a lot of control. These bilateral deals between countries and Chinese vaccine producers are often not transparent. That is a frequent criticism. And clearly countries which are in the Belt and Road Initiative have accelerated access to vaccines. So some people complain about a mix of commerce and soft power. And they also complain about a lack of transparency, particularly in pricing. So for example, the Sinopharm vaccine it's not a cheap vaccine. In Hungary, it sells for $36, and in Senegal, it sells for $19. But it's a little bit hypocritical to complain about China in this context, because that is something we are seeing from every Western vaccine producer. There's no transparency in pricing. We have very little understanding about the capacities, what is happening, who is getting how much vaccine. So that's not something where China stands out. There's also a feeling that there's no unified approach. Individual companies are negotiating. Yes, but that's the same with Big Pharma, isn't it? Another problem that China is now increasingly facing is that they would like to do large phase 3 studies, particularly with new candidates coming out. But it is a massive problem doing this because they have not enough COVID cases in China to do a vaccine trial. I mean, they are so sparse, these cases, and then only for such a short time that it is probably not possible to do any phase 3 study there at all. The other problem is that once a trial has been undertaken, that was last year the case, there was a lot of insistence of complete control from the Chinese partners, which is difficult to do if you do a trial outside your own country. So that brings us to this big challenge, which for all vaccine producers now, which is that it has basically the window to do placebo-controlled trials is closing. But what I mean by that is that if you want to randomize a trial and give half of the people a placebo and give the others the active vaccine, that means half of the people are unprotected. Usually we are trying to vaccinate people who are at the highest risk because there's the highest incidence. That means you get the cases quickly. You finish you can answer your question in a short time. That is becoming close to impossible because the very few people say, well, if I can get a vaccine, why should I take the risk of getting a placebo and take the risk of becoming infected? That's one thing. Ethics committees, which have to review every single trial and are examining in great detail, are not happy with the word placebo. They are not happy with using placebos. So I think it will be very, very difficult in the coming weeks for the next wave of vaccines to do placebo-controlled trial. So what can they do instead? One of them is to do non-inferiority trials. What I'm talking about in a placebo trial is a superiority trial, that the vaccine is much better than using nothing, than using a placebo. A non-inferiority trial, I mean, is using an established vaccine which is already out there and compare it against the new vaccine that is coming out now, and compare these two vaccines. The vaccines, it's not as clean because we know that the established vaccine may also cause adverse events, may also cause reactogenicity. So you don't get a clear picture anymore. You just have to hope that you have understood the first vaccine really well, the prototype that is out there, the established vaccine that is out there, and then make conclusions based on that. But a lot of that is kind of this inference, and it's not quite as clean as doing a placebo-controlled trial. And then if people don't want to do this, there is a particularly clever way of addressing it, and that is bridging studies with neutralization antibody titers. And I would like to explain this, if you give me a second, how this is done. So basically, when a person has COVID disease and recovers from it, the convalescent person has antibodies. And we can measure the titer, the density of these, the level of antibodies in the blood of that person. And then we can take the blood of somebody who is vaccinated and measure the antibody titer in that person who has been vaccinated. And then we can take the antibodies from these blood from these two people, from the convalescent person and from the vaccinated person, and contain the titer the density, again, the level of antibodies from the vaccinated and from the convalescent, in the convalescent blood, and then make a comparison. This is, if you can make it work, it is a very, very elegant, fast, affordable, cost-efficient method of doing it. The difficult part is to get the correlation correct. And a lot of people are spending a lot of time the last month trying to use the data that they have from the vaccine studies already, seeing blood serum from people who have been vaccinated, examining the titers and comparing it to convalescent titers. That would be a very elegant way of doing it. And I would like to show you some of the ways this is done here. Just bear with me here, please. So this is for the Novovax vaccine, the vaccine I have mentioned earlier. This is the subunit vaccine, which had in clinical trials showed 95.6, 96% protection. And when we look at the antibodies produced by people on day 0 by the time they are vaccinated, of course, there are no antibodies because that is, they have just received the vaccine, or they will just receive the vaccine any minute now. And then 35 days later, they have an antibody titer. And here the antibody titer is 4-fold higher than a convalescent titer, which you see in blue here. If you allow me to show it here. So this is day 0. This is day 35, and this is the convalescent titer. And then we can make an estimate that this titer is about 4-fold higher. And it stands to reason that this person is at least as well protected as somebody who has had the disease already. And then you can use the same approach for all the vaccines which are out already, basically, and that has been done. And then we find here that for the mRNA vaccine from Pfizer, which has 95% protection, affords 95 protection, the antibody titer is 3.8-fold higher than a convalescent serum. For Moderna, it's 3.4. For the Chinese Sinopharma, the one with the 79%, that is about comparable with the convalescent titer, so it's about the same. Oxford is the same as Sinopharma, but they are about in the same range. So the earlier estimate was 62, and as we heard earlier today, it's probably 76, maybe 79% protection. So that fits nicely here with these titers. The Jensen vaccine is 2.3-fold lower than a convalescent titer, and Sinovac is 6-fold lower. And again, here, Sinovac has only in trials found to be 50% protective. So these neutralization antibody titers have a very good predictive value what the efficacy in a large trial will be. And if we can't do these trials anymore for a number of reasons, then using these neutralizing antibodies are a very attractive approach towards it. The way earlier today there were long discussions again how this can be, how the regulators should react to it, how it should be approved, and it will probably come out that you still have to give the vaccine to a number of people to show that it's safe. You can do then in these people get the antibody titers, and then once the vaccine is released, do an effectiveness study afterwards. Particularly if a completely new vaccine should come out, then and it's the emergency authorization is based on neutralization antibodies on correlates, then there may well be a need to do an effectiveness study, which is basically you vaccinate everybody and then look how many of these people, how many people who had disease after vaccination have been vaccinated and how many have not been vaccinated in a community. I hope I made this clear. I hope I was able to explain that. Okay, let's move on. How many COVID vaccine doses have been administered so far? And here we break this down by country, not by vaccine. And clearly the leader as of March 22nd is the United States. If you're interested in this, by the way, there's a fantastic website called Our World in Data, which shows these data, and they are updated more or less every day. Followed by China. As I said earlier, China is exporting most of the vaccines. So these are only doses administered within China. And India is then number 3. The United Kingdom has done very well, has caught up from the rather disastrous last year and is doing extremely well this year. The European Union is down here and is, I mean, there 10 million people are vaccinated, but people living in the European Union are at the moment quite disappointed with what is happening. And there is a lot of unhappiness apparently. So that brings me to the next question. How many vaccines do you actually have to administer until you can interrupt transmission? And this is a question which comes up so often that I would, if you don't mind, quickly like to go through this with you. So the vaccine coverage required to provide immunity protection, this is herd immunity. So by that, I mean is if you only have vaccine and no other measures, how many people do you have to have vaccinated? And there is this equation here, which really gives you a very concise answer to it, to this question. And this is 1 minus 1 over R naught. And R naught was always in the headlines during the last year. We don't read so much about it anymore. R naught is meant to be the expected number of cases directly generated by one case in a population where all individuals are susceptible to infection. So to interrupt transmission by vaccination, and by vaccination alone, if the R naught is 3, which we thought it was last year, kind of perhaps the upper limit, then the vaccine coverage needed is 1 minus 1 over 3, which is 66%, right? And if we deal with variants which are more transmissible, then the R naught may increase to 4. And here the vaccine coverage required is then 1 minus 1 over 4, which is 75%. So if you look at this here, we need 75%, and this is 75% of the complete population, not from subpopulations. From the entire population, 75% have to be vaccinated. So if you exclude populations and they are not eligible for vaccination, you have a major problem. They are of course pregnant women, but pregnant women are only 2 to 3% of the population at any one time. If you exclude children, you exclude, for example, in the United States, a quarter of the population. In very many low and middle income countries, this population is even bigger. So if we don't start very soon, vaccinating children, our hope to get by vaccine alone down to a level that the transmission of COVID is interrupted is not going to happen. And then the other big problem, which is of course a luxury problem, is vaccine hesitancy. So knowing what the coverage should be, what is the coverage on March 22nd, according to our World in Data? Israel has already vaccinated more than 100% of its population. I mean, most of these vaccines are 2-dose vaccines. That explains to the 112. United Arab Emirates are doing very well. Chile are doing well. And United Kingdom has performed extremely well this year. European Union here, down here, is not doing so well, but it is vaccinating. Bangladesh, you could say, well, only 3%, but now compare this to other places, to the entire globe. If you look here at a map of the entire world, then you have places here like the United States, which are dark. That means there is vaccine coverage taking place. Europe doesn't look bad at all there. There are some places in Asia, here Southeast Asia, which are not doing so well. But the big problem, clearly, the elephant in the room, is in the middle, Africa. And there are no data because there is very little vaccination ongoing at the moment. That here is the big problem. So what is done about vaccinations in Africa and other low and middle income countries? And for this, the COVAX facility was created in April 2020 by the WHO, the European Commission, and the government of France. And now the pillars which are carrying COVAX are UNICEF, Gavi, WHO, and CEPI. By last July, 165 countries representing 60% of the human population had joined COVAX. And based on the data that I could find, the COVAX at the moment has 240 million doses of the Serum Institute of India version of the AstraZeneca Oxford vaccine and 96 million doses of the Oxford vaccine produced by AstraZeneca. And 1.2 million doses have been donated or are available from Pfizer BioNTech. I shouldn't say was donated, it has been pledged. And we know that 10 million doses will come from China. So looking at this data, it is pretty obvious that the majority of the vaccine doses will be this AstraZeneca vaccine. And the AstraZeneca vaccine will be absolutely critical for vaccine coverage in low and middle income countries. These vaccines I try to illustrate here. So there is one stream coming from India, from Serum Institute of India, from AstraZeneca, and then we have here vaccines coming out of China. That is the other stream of vaccines that will provide vaccines for Africa. Then let's move on to variants of concerns and escape mutants. What's that, a variant? A variant is a subtype that is genetically distinct from a main strain, but not sufficiently different to be a distinct strain. One of the big problems with variants is that they have cropped up at the beginning of last year and there is no unified nomenclature for them. So if you talk to somebody in the United Kingdom about a variant, they probably mean something else from somebody whom, when you talk to somebody in the United States, who is talking about something else, somebody in Brazil or Japan. Basically every country has initially used their own name for these variants. And unfortunately, the only name that really sticks is the country name. And that is unfortunate because, for example, the most sequencing has been done in the United Kingdom. And for example, Denmark has done tremendous amounts of sequencing. A large number, a high percentage of the viruses that have been found there have been sequenced. Now, if you name the variant after the country, you punish the people who are finding it because the more you look, the more you find of these variants. And that is not a smart way of doing things. So what happens now usually is that people say, for example, this is the B117 strain, which comes from the United Kingdom. Then everybody seems to understand what that person is talking about, but it is a little bit complicated at the moment. Here, I'm just trying to show this, that the way these variants emerge and spread is complicated. Just to give you an idea of the flow, here, this is the original strain, the ancestor, so to speak. And then from there, it goes down here into Europe and becomes the B1 strain. And then here has all these offspring, of which only the ones here with these squares are of particular concern, particularly because the red square indicates that these variants are associated with antibody resistance. That's antibodies from convalescent or antibody from vaccines. And the turquoise squares are associated with, that indicates strains associated with increased transmissibility. So you have here this strain, obviously, and then you have here these ones down here. And then the big ones from Brazil, the P1 and P2 strain down here. So this is just to give you an idea of the complexity of these variants as they are emerging. Why are they so problematic? As we heard, they are spreading faster. They increase their R naught. We need a higher coverage of the vaccine, as I said earlier. There is a risk that they could escape vaccine protection. So if you think of a scenario where more than half of the population is vaccinated, clearly a mutant, a variant strain, which can escape this protection has a huge survival advantage and will spread much faster and will dominate the field. In general, variants are not more lethal or pathogenic, because if they kill the person that they have infected, that person cannot transmit the virus any longer. The same if it's highly pathogenic, the person is immediately bed-bound, quarantined, cannot transmit any longer. So that is not in the interest of this virus, basically. The optimal virus is a virus which is not harmful and can spread very fast without causing damage. How should we deal with these variants? So the first step is that countries are making already large investments in the collection and the sequencing of SARS-CoV-2. Then there is a need to continuously assess the protection afforded by the prevalent variants of concern. And this can be done with these neutralization assays I was referring to earlier. The next step has to be, if we find that the vaccines are no longer protecting against variants, is to adapt the vaccines against these novel variants. The thought is, of course, the first thought is we should replace the vaccines. But another idea which may be also quite useful is, as long as there are still some of the so-called old strains in circulation, is to make vaccine cocktails, that is polyvalent vaccines. And the ultimate hope there is that if you have enough targets on the virus, then basically if you have 2 or 3 vaccines in a cocktail, the mutant has to make so many mutations that it is no longer viable. So there is a limit to it. So we don't have to think that it goes on in infinitum, that you always have to add a new variant. There can come a point where the virus can no longer propagate. And this is not such a completely unusual scenario. Actually, we have something similar happening with the flu vaccines. Flu vaccines are changed on an annual basis. Sometimes they are completely replaced. Sometimes we add a strain to it, depending on what is in circulation at that, or is predicted to be in circulation the following year. So there is a mechanism how to do this, how to adapt vaccine to new strains. And vaccine developers will have to work very closely with the regulators to develop a mechanism how that can be done in an ongoing fashion, because clearly you do not want to go through phase 1, 2, 3 studies each time you adapt the vaccine. There has to be a very quick and efficient way to do this. Otherwise, there is no point in doing it. If we need a year to develop a vaccine, by that time there will be already a new variant in circulation. Then just a word about vaccine hesitancy. This is the other big problem to vaccine coverage, obviously. It is an old problem. There's nothing new about it. People refuse to be vaccinated. It happened with the first vaccine, smallpox, already. There was an uproar in parts of the population. But what we're seeing here with vaccine hesitancy is, I think, a little bit unusual development. We are approaching here around 2000 or so. And there something really changed. It went up more exponentially. And then, of course, last year we had a lot of references, a lot of people talking about vaccine hesitancy. Sorry, I didn't make this clear. Here along the y-axis, we have the number of references to vaccine hesitancy. Why are people hesitating to be vaccinated? There are a range of reasons and a range of reasons why people are doing this. There is a completely irrational fear of side effects and saying, well, wait a minute, let's see how other people are doing before I get vaccinated. There's completely irrational decision. It's not a very altruistic point of view, but it's understandable. Then there are a large spectrum of people who have different belief systems and feel that the people who are suggesting that you should be vaccinated may not have their interest in mind, first and foremost. This is particularly the case with minorities. And then we have a small group of anti-vaxxers, and it would be a mistake to mistake all people who are hesitant with anti-vaxxers. Anti-vaxxers who are fundamentally against vaccinations is a very, very small fraction of the people who are hesitating to become vaccinated. But having said all this, this is really a luxury problem. At the moment, I think working in low and middle income countries, we want to have access. We are not worried at the moment that some people would not want to take the vaccine. Correct me if I'm wrong, but at the moment we are trying to get access. And once we have that and we have managed to get a third of the population vaccinated, then I think then then we have to work again very strongly to get the rest of the people also pulled in and vaccinated. So coming to an end here, I was asked, what are the long-term effects? What is the transformative effect of this pandemic? And is there a transformative pandemic on the biosciences from my perspective? And I think there's a huge effect. All of us who have children or have lived with children over the last year, they know that this has left a lasting impression. And I have no doubt that my children will talk about this in the future and tell their children and will tell their grandchildren about it. And it has done something in their mind that they think in a different way about the world. So what's coming out of this is that they will vote for people who have a different attitude to pandemics. What is the point of having a fantastic military, having a great defense if you get laid down if you get killed by a pandemic? So I think people will vote in this direction. We have seen this last year that President Trump was voted out. There's a broad consensus that it had something to do with his reaction to the pandemic, and other people will pay attention to it. So there is, from my perspective, little doubt that in the long term, there will be an influence of policy. People will keep that in mind, that this should not happen again, and will make investments accordingly. So that's one big change that I'm expecting. Technology breakthroughs. I talked earlier about this astonishing mRNA vaccines and the success of vectored vaccines, which 20 years ago, nobody would have believed in. As I was trying to say, the mRNA vaccines have something a little bit miraculous for people working in vaccines. And that is a technology which will become available much more broadly now, that will be used for a lot of diseases other than COVID. Vaccine targets which have not worked in the past, I'm sure will be readdressed. And we can expect considerable progress in biomedical technology in the future coming out of this. Then capacity and resources. So a lot of people have now been working on vaccine development, have worked on vaccine production. There's a completely new generation of people working on vaccines who suddenly had resources like never before to work on these COVID vaccines, and hopefully can also work on other vaccines. And then we have technology. 20 years ago, I started working at the International Vaccine Institute in Seoul, Korea. We had real problems getting a pilot plant going, which would have costed about maybe $100 million. It was ultimately not affordable. Now, I think there is much more technology available. There are these huge facilities to produce vaccines later on. So I expect that the world of vaccines is changing, and people who are interested in this, young people, can get jobs there. They can learn how to do this and become vaccine developers. There's a complete new area an era opening up, I would expect. And as I was trying already hinting at is, we have not been very successful, for example, with malaria vaccines, not to mention tuberculosis, HIV, etc. But these vaccines targets, I think, will be in the coming months addressed again. At the moment, people are so involved with these variants and adapting the COVID vaccines to the variants that there's no time to think about it. But once we get out of this, the battle, I would hope that a lot of interest will be focusing on other vaccine targets. And then this is the last slide. So what are the lessons, implications for other diseases? In the short term, it is negative. There are immediate funding decreases. We received yesterday a letter from the United Kingdom Research and Innovation Board for the official development aid, and they have drastically reduced their funding. Funding that was already pledged has been pulled back. So that is going to have very negative effects for people like me working in global health. Then, of course, people in the countries know exactly how strongly they have been affected by this pandemic. tourism, remittances have gone down, exports have gone down, massive changes. And then, of course, there is the character of our collaborations, which is completely changing now because people haven't been visiting for a long time. And suddenly we find out that we can do a lot more communication online than we had thought before. So in summary, let me repeat, I think a lot of people will be working in biomedical research and public health. There are enormous opportunities of opening up, and the application of these new technologies will bring progress. Thank you very much. That's the end.
Yeah, so thank you very much for that thorough and extensive session. And we have lots of questions for you already, lots of questions. They've been coming in severally, even from the moment you started the presentation. But before we go into these questions, one question that I would ask you is this: why do you think we hear so much about vaccines from Moderna, the Pfizer, and AstraZeneca, while what is happening in China is not getting much attention? Then looking at the trend and how the pandemic and vaccine rollouts are all going out across the world, what do you think is still the long-term plan? How do you think we can achieve equitable access to vaccines across the world such that almost every country, or let's say every country, can achieve these desired herd immunity by mid-2022, if that is possible? So quickly answer those questions before we go into the lots of questions that we have for you.
Thanks, Paul. Why is Moderna, Pfizer, and AZ, AstraZeneca dominating the field is because there is a very kind of blinkered view from the international press. It's even more extreme that if you follow newspapers from the UK compared to USA, compared to Europe, there are huge differences, and the way they are reporting, particularly about the AstraZeneca vaccine, is highly variable between these countries, obviously. And there is, the word nationalism is coming up time and time again, and it is so sad to see that in this age, in this day and age, people are still so jingoistic about their products. So that Americans very strongly feel that Pfizer and Moderna is their product. The people in the United Kingdom think that AstraZeneca is their product, that is their vaccine, and they are really proud of. And then that it goes to the degree that in the European Union now people are just saying we don't want the AstraZeneca vaccine. I mean, you know, the people who should be vaccinated says, no, we would like to have the other one, just based on these rather irrational perceptions. It's really sad, and I mean, it's just I put it down to nationalism. Very single-minded. And then how do we get more equitable access? That's the big question. I'm optimistic. I showed you at the beginning all these vaccines that are coming out. I can see the numbers of vaccine that people are planning to produce and actually going about to do it. There are setbacks, like for example, then India later today decided to reduce their exports or want to block them for a while. But there is so much vaccine coming out from so many places that I think we can be optimistic that in the coming month, not as early as we wish, but there will be maybe a glut of vaccines. So there is, I mean, nobody needs more than 2 vaccines in a year, right? I think we agree on that. And nobody wants it. So once you have these billions of vaccine doses, at some point, somebody is going to do the right thing and say, well, let's send them to the people who are not being vaccinated. And I mean, it is so fundamental that if there is somebody left who is not vaccinated, if there's a whole population that is not vaccinated, nobody can be safe. It's just so silly to think that you are protected because you are in the United States or in the United Kingdom and you don't have to worry. No, you have to be worried, because if you don't leave, and even if you don't come into contact with that person, somebody of your tribe, somebody of your village, somebody of your city will travel and will become exposed and then brings it back to you. So you cannot avoid this. The only way you can feel safe is if everybody is vaccinated.
So since there are lots of questions, Gus, just if you can go back and forth, Mrna, yes, for these series of questions. So the first question we have is from Mrna. He said, regarding the talks of continuing mutation of the virus, how far would it alter the produced vaccine's efficiency. So how do you think mutation would impact vaccine efficiency in a nutshell?
Yeah, it is pretty unpredictable what is going to happen. So we see already that in the trials that are ongoing, the protection is less than we had hoped. So it probably makes an impact. It doesn't make the vaccine useless, but it reduces the protection. So instead of 95%, it becomes 85%. That is what I think is going to happen. And so it's a question of continuous monitoring.
Oh, okay. So Christian Castro is asking, why not talking about inactivated platforms? Because they have wide use in developing countries that they don't require special storage. So what do you say to this?
Inactivated vaccines, that is the vaccines from China in particular, and the Bharat vaccine from India. So that was on my graph there. These the ones at the bottom, Bharat, Sinovac, Sinopharm. That's all inactivated vaccines, and I think they have a number of strengths. It's a technology which is around for a long time. People are familiar. Vaccine producers are familiar with using it. They have made lots of experience with it. They know how to do this, and they know how to do it well at high volumes. And I think one of the core strengths is that it offers so many vaccine targets. It's a more robust vaccine than using just one little protein and hoping that that immune response will protect against variants. So it has advantages. I think what is one of the big advantages why people in USA and Europe are not so fond of this methodology is because basically you have to brew up these huge bioreactors of virus to make it, right? So you have to produce this COVID, hundreds of liters of it, which is a huge hazard. If that goes wrong, you have a really big problem. So it's a more old-fashioned way of doing things.
Okay, so Vinod from
please go ahead, do it, use them, bring them on, please.
Okay, yeah. Vinod from Facebook is asking, people who have taken vaccines are also getting COVID. We need to understand what protocols need to be followed even after getting vaccinated. What do you say to this?
Yeah, even the Pfizer vaccine is 95% protective. So that leaves 5%, right, of people who can still become infected. And for the Sinovac vaccine, it's about 50% protection. So half of the people who have been vaccinated are still at risk of becoming infected later on. They don't not necessarily become infected, but they remain at the risk. But I think there's one important thing which I didn't make clear, which is really important to understand, is that even if you become sick after a vaccination, the chance to become so sick that you have to be hospitalized or that you die are drastically reduced. So with the Pfizer vaccine, that hasn't happened. And I think it's still true with the Sinovac vaccine, is that very few people, if any, in the trials who have been vaccinated that died. They became infected, they got mild disease, but they did not get progress to severe disease and death. And this is all I want, isn't it, for myself? I mean, I want to survive. That's really the important thing. So for me, there's a really good reason. If I can get that vaccine, I take it because it increases my survival chances dramatically.
Yes, so Cyrus Sivat is asking, what about the vaccine from Cuba?
From Cuba? Yes, Cuba. The Finlay Institute. Thank you for asking that question. The Finlay Institute has developed 3 vaccines, not just one. I mean, you know, Fidel Castro's big thing was vaccines, right? So he had this as his private enterprise, was this Finlay Institute, where Finlay Institute scientists got whatever they wanted from Fidel Castro. And it still seems to be functioning very well. And they have now 3 vaccine candidates out. They are all subunit vaccines, the way I understand it. And I think they are going into trials this year, later on this year. They have also very beautiful names. Sorry, I don't remember them. They are very poetic. He gave them poetic names, their vaccine. So they are enormously proud of their vaccine skills, their vaccine development skills. They are making vaccines, not one candidate, but 3 candidates. They have a good chance of coming up with their own candidate. And they go as far as that they hope that they will be in the future able to export the vaccine and make money with it. Yes, Cuba.
Okay, yeah. So Yeah. Isa Friedman from Facebook is asking that to what extent, if any, would annual flu vaccines be compatible or clash with the efficacy of COVID vaccines? And would the COVID vaccine eradicate the need to take the annual flu shots?
So at the moment, there are multiple parts to this question. The first one is there has been very, very little flu this year because people, for the first time, use really social distancing, hand washing, etc. So that has collapsed the transmission of flu vaccines this year. So it's not a thing to worry about this year. Next year is, it may look different and in the future. To know whether you can give a flu vaccine compared with a COVID vaccine, you have to do a trial. You have to see what happens because there is no way you can foresee the future. But based on what we are knowing, there's no reason that there should be an interaction between the two vaccines. And I would be quite happy, and I will, once I am vaccinated against COVID, will take my flu vaccination. I continue to take that because it's just a triple health not having the annual flu episodes. So I mean, if you think about it, let me think about it. Allow me to put it to you in another way. You get now COVID and 3 weeks later you get flu. That can happen to you. It's bad luck, but it can happen. So you get first an immune push from the COVID, then you get the flu, and you're going to build antibodies against both of them. And then you're going to be immune for the coming months against both diseases. So you can do the same with a vaccine. There is a priori no reason to think that this causes an immune overload or some interaction. So I would be really optimistic about it and risk it.
Yeah, so Miriam is asking, is there vaccine hesitancy in China? And also, isn't it concerning that so many people outside of China are getting the Chinese vaccines without phase 3 data?
Yes, that is a concern. We would really like to have more data. We would like to have published phase 3 studies, these randomized controlled studies. I'm hoping, I'm optimistic that we get more data from China. But you know, a year ago, we were hoping kind of last summer that China would use this opportunity to open up to, because they have made very good vaccines in the past. They can do this, but they have not entered the international market at the same level as Indian vaccine producers have. So there would have been the opportunity to show the world we can make these vaccines and we can make them the same way you are doing them. We can make them at least as well as you are making vaccines. But unfortunately, they have not used the opportunity and keeping this very strict control over it, making sure that there is nothing unexpected happening with which they cannot later on control. And I mean, if you do a big international trial, and you have a data safety monitoring board, it will happen to you at some stage that the data safety monitoring board, if it is independent, says, stop, we have to pause this trial. That has happened with most of the trials that are going on. And you have to accept that the international data safety monitoring board, if it is independent, will say this to you. And unfortunately, China has not used this opportunity to go through this experience and allow the world to see how good their vaccines are.
So Linda Lee is asking, considering the advantages of the inactivated vaccines that you mentioned, and she's not asking, does this mean that Chinese vaccines which use the inactivated platform could be more effective against variants than Pfizer and Moderna vaccines? And how can mRNA vaccines deal with the variants?
So that is speculation. I mean, it's just from the way that the vaccines are made, that it is the entire virus, not just a protein. That's I think I explained this. How do you deal with it is that the BioNTech, the people who developed the vaccine, which Pfizer is producing, said already last summer when the first inklings came along that there might be variants going on, that within weeks they can reformulate their vaccine. I mean, they were ready with their vaccine basically in April last year. It took them, I think, less than 3 months. So they have done it now within weeks. I would say 8 weeks. They have a new vaccine basically ready. So they just have to then produce it and deliver it. That's one thing. That's the production. But what is enormously time consuming in vaccine development is also the regulatory process. So there is a need that the regulators are working together with the vaccine producers continuously. So on the first day when BioNTech is going to make the new vaccine, that they say, here, this is the blueprint of this vaccine. This is what we're going to do. Let us know what you want to know, that we answer your questions in a timely fashion.
Now,
That's the way.
Betty Freshsome is connecting vaccines development with the role of journalism. So I really would like you to take this question. The question is this: a study just showed that the news reports are overwhelmingly questioning the safety of vaccines and highlighting potential problems. Is this the correct way to inform the population about this situation? And is the glass half empty or half full at this time?
Yeah, that's a good question. And of course, I don't know the answer because I'm not a journalist and I haven't thought about these problems to the level that you have thought about it. So for me, it's a little bit difficult to answer it. For me, it's very inconvenient and uncomfortable when I open the newspaper and see there's another bad news, because I'm very concerned with vaccine hesitancy and the material that it gives to people. But I accept that it is essential that people have to know that they get open information, that the information is out there. But of course, it is out of proportion, like these now 2-3 days of AstraZeneca news headlines in the international press every single day. This is probably not justified, and I don't know how many people are interested. But I mean, you journalists know much better what your readers want to see, and if readers want to see that, that is fine. I mean, it's part of the process how we are dealing with things. I think I would feel a lot less comfortable if there would be a suppression of news. That would worry me a lot more, to be honest. Does it make sense?
If I would, yeah, but yes, I want to take it a little bit further. So from the perspective of a scientist, from the perspective of a researcher, that you know how vaccines work, how vaccine researches are conducted, what do you think journalists can do differently with the way they cover vaccines? And what are your major concerns concerning the impact that the current vaccine reportage could have on current or future vaccine initiatives? It's a total opinion question. So,
yeah, I would like to take a step back and say that I don't think that I know enough and everything about vaccines. I'm learning about vaccines, and I haven't learned so much about vaccines as in the last 12 months, and I'm probably not the only one that has happened to. It was incredibly, and it remains an incredibly fast learning process, quick learning curve. And I think all we can do is address at various levels. It's because people have such a huge spectrum. So some people have to be addressed in one way, other people in another way, that that we take the time and really explain what we know about vaccines and what we think about it. And that includes our concerns. If there's something that we are worried about, and say, well, I would rather wait for a week, or maybe another vaccine that I think is also acceptable, that there is no pressure on anybody that they have to do something, that you have this openness and the freedom, and hopefully also the freedom to choose between different vaccines at some stage, that you can say, this is what I would like and not to do, and provide people with enough information that they can make this decision for themselves. I think the idea of forcing people to be vaccinated is really kind of something that doesn't agree with me at all.
Now, so what is your opinion regarding, I'm trying to bring Vino's Facebook question to make it more relevant to the discussion. So what is your opinion regarding development, comparing vaccine, COVID-19 vaccine development with COVID-19 treatment development? So what can you say regarding these issues?
That's a very, very good question, because again, I don't know enough about it. At the beginning, in the first half of last year, it started off with the focus was entirely on treatments. And then there was not much happening in terms of treatment. There haven't been many really overwhelming successful treatments which left a big impression. There were a couple of drugs which came along one after the other, but none of them really was an overwhelming success. The only one which sticks out now is dexamethasone, which is an old drug repurposed, and basically an anti-inflammatory drug, which is a certain stage of the infection, if it given in a very artful fashion, can do wonders and can save life. It's not at the beginning of the infection, but at a later stage. If it's done well, it's fantastic. It works really well. Besides that, there are not so many good drugs have come out and really had the success that you see from Pfizer, from vaccines, 95% protection. That we don't see this. We don't have any antiviral drugs which really kind of kill the virus like this and reduce this infection immediately. And we are not so good with antiviral drugs, full stop. I mean, remember how long it took until we developed drugs to deal with HIV. And that's also a virus. And so there's a similar situation. I mean, there are still trials ongoing. Our group works on chloroquine prophylaxis. And that is extremely difficult now to do any chloroquine prophylaxis trial because there are so many vaccines out there. And people prefer to use a vaccine to a drug. So again, testing prophylactic drugs has become very, very difficult at the moment. Therapeutic drugs are in development, but there have been no breakthroughs which are comparable to the vaccines.
Thank you. So let me connect two questions now. We have one that says, how long does it take in terms of acceptance? Does it take for long-term adverse effects to become noticeable? Adverse effect of the vaccines. How long does it take to become noticeable? Then Kanchan Stava is asking this question, that what could be the possible reason behind getting COVID after 2 doses? Or unrelated death? Quick background to that question. The person wrote, in India, several people have caught COVID-19 even after taking both doses. This has raised some concerns among those who wish to take vaccines but are apprehensive. So a few deaths have also been reported, though vaccine makers said it is unrelated to vaccination. So, so that's the question, and that's the background.
Yeah, so we had one on adverse events and one on vaccine failures, right? That's the two things. So if I can combine them, if you allow me to put this into one answer, try to answer it in one thing. If you think about it, you know, we are vaccinating 100 million people, say, and we want these 100 million people. We don't want them to be that they have a too strong immune response. If the immune response is too strong, it causes damage. If it is too little, then there's no protection. And so you can imagine that with all these biologic systems, it's kind of this Gaussian distribution, you know, kind of, you know. So there are some people at the extreme where the vaccine doesn't take, they don't react to it, right? And the huge majority of people to them who have no take, you will never find out because they are not exposed and they not become infected. But the few where the vaccine did not take and they have been exposed to it, they become infected. And they are unhappy, and they are very unhappy customers, and say, how can that happen? Yeah, it's as I was trying to say earlier, it's related to the protective efficacy of the vaccine, and this percentage is higher with Sinovac than it is with mRNA vaccines. We know that. So that's the first part. But then, how do we find out about the other extreme if the vaccine causes an adverse event in somebody? And this is the idea of pharmacovigilance, that after people are vaccinated, there should be a system that if they have any complaints, that they have a number, that they have a way to report this, that this report is correctly and immediately transmitted to a central point, and that should always be the vaccine producer. They have to have the responsibility to collect the data. They are supervised that they are doing this responsibly. They cannot hide it. And my experience in working with vaccine companies, they are really strict about this. So they get the information, they immediately process it, have to process it, and then take the correct steps afterwards. And if they find, for example, that there is a clotting problem, which is much higher than would have been expected in the normal background, then they have to take their own steps and say, stop, we have to pause until we understand what is going on here. For example, whether there are many more cases suddenly coming in of this complaint, we have to wait until we restart vaccination. That's the idea of pharmacovigilance. And we had many decades now time to develop and refine these systems. They are still not perfect, but I think the blood clots in Norway show very clearly that there is something in place, that these things are picked up, that people react to them. They see them in Norway immediately within days. They know that there's something happening in Austria. Then people compare this to the data in Germany, and everybody says, well, let's stop anything and compare our data, work our way through the data before we continue vaccinating. That's I think how
these are. Yeah, so thank you very much. We've really gone way beyond the time allocated for this session. And it actually shows that this is an interesting topic that journalists are interested in getting more information. And we believe that you've been able to communicate and inspire our journalists to identify potential news story angles and to have factual information that will guide them on reporting issues regarding vaccines, especially as it has to do with the COVID vaccine. And I really want to appreciate everybody that is on this webinar today. So thanks so much for joining us. I hope this webinar has informed you better on the topic of COVID-19 gray areas. And importantly, it helps inspire potential new angles for stories. So if you're not a member of the ICFJ Global Health Crisis Reporting Forum yet, I encourage you to please find the group on Facebook, or for more information about the forum, visit www.icfj.org. And if you would like to access journalism resources on COVID-19, please check out the International Journalists Network at www.ijnet.org. And please don't forget to fill out a survey coming to your inbox after this webinar. ICFJ would love your feedback to improve programming for this series. So apologies to everyone that we couldn't take your questions. We will get the questions across to Prof, and we would attempt to communicate his response to you. So thank you very much for an amazing time.
Right,
we are going
thank
to
you.
Yes,
thanks,
so thank you very much.
Bye.
Yeah, I really appreciate it. Bye, everyone. So we are out in 5, 4, 3, 2, 1. Bye.